Today, I would like to talk about our research results for utilization of advanced drug delivery formulation. I will allow time to describe our recent preliminary achievements also. Please focus on how we design these drug delivery systems for biomedical application. Biomineralization is a natural process that living organisms use to make minerals such as calcium phosphate or calcium carbonate in order to strengthen soft tissues. Especially this process is very important to formation of bone and teeth. And in nature, there are also minerals from mines and soils like rhodochrosite, siderite, copper arsenite. In addition to the robustness character of minerals, this mineral structure is influenced by pH level and the dissolution rate changes. Especially in the case of calcium carbonate, the solubility increased with acidic pH, and the process makes carbon dioxide. For example, at the physiological pH, the solid calcium carbonate network is preserved, but within the endodermal or tumoral acidic environment, pH level is much more acidic, and the calcium carbonate mineral network dissolves with generate calcium dioxide gas. For using this calcium phosphate and calcium carbonate minerals, we successfully developed the various nano carrier system, and very recently we have choose the manganese carbonate or ferrous carbonate phase for the biomimetic mineralization application. Using this mineralization process and technology, we have developed a multifunctional mineralized delivery system for cancer nanomedicine, such as chemical, drug, protein carrier, nitric oxide carrier, and oxidative stress amplifier, and contrast imaging agent. Next, move on to our working basis. So as a basis study, we reported the calcium carbonate mineralized nano formulation based on black copolymer templated mineralization. The black copolymers of polyethylene glycol copolymer polyaspartate was used as a template for mineralization. Anionic polyaspartate serves as a nucleating template or calcium carbonate mineralization. The consequential binding calcium ions and carbonate ion induced the supersaturation and thus result in the formation of mineralized nanoparticles. The addition of drugs is also optional. And next, after the successfully making the drug-loaded mineralized nanoparticle, now I would like to talk about the working principle of this nanoparticle. Recall that calcium carbonate nanoparticle maintain their robustness structure at physiological pH, like a blood vessel. But at the tumoral acidic pH level, calcium carbonate mineral nanoparticles start dissolving to relieve carbon dioxide and produce gas bubbles. So we can use these bubbles. So we can use these bubbles to generate ultrasound echogenic enhancement to monitor the nanoparticles at the tumor area. As the calcium carbonate produce carbon dioxide gases, Simultaneously, the loaded therapeutic drug will also be released. In this way, we can both diagnosis and treatment at the same time after accumulation of particle into the tumor site, and therefore therapeutic is possible and realized. And next, we have identified the effectiveness generation of gas by measuring the mass of carbon dioxide at the tumoral exit pH. And we demonstrate the micron-sized bubble formation at tumoral pH. The generated carbon dioxide molecules coalesce to form the large size of bubbles, and it is suitable for ultrasound imaging. At the in vitro release profile experiment, the calcium carbonate mineral nanoparticles strongly inhibited the drug release at the physiological condition at pH 7.4, and however, selectively released the drug at the tumoral pH 6.8 that was confirmed to promote the drug release by dissolving calcium carbonate mineral. It means that the correlation between the pH-dependent calcium carbonate mineral dissolution and toxo release profile. This slide shows the pH dependence of in vitro ultrasound imaging. Contrarily to negligible signal at pH 7.4, we have found significantly enhanced the ultrasound contrast signal at the tumoral pH 7.2 to 6.8. It is interesting to find that the weakly acidic pH of minute different from pH 7.4 can generate the substantial signals. We can expect that the lower pH rapidly dissolved the nanoparticle and produced a large amount of carbon dioxide bubbles at the short time period and fade away. This slide shows the preliminary in vivo ultrasound imaging data after injection, after intratumoral injection. The liver was used as a control, soft semi-solid tissues. We measured the pH of the liver and SCC tumor cancer tissues with the semi-solid electrolyte, and found that around pH 7.3 and 6.7 respectively at the liver tissue, there is no variation in the ultrasound signals. On the other hand, at the tumoral tissue, the injected the nanoparticles generate gradually the enhanced ultrasound contrast imaging. So next study is about the in vivo efficacy of total vision loaded calcium carbonate nanoparticles. After confirm the in vivo ultrasound imaging, we compare the tumors after injecting the toxo-loaded mineralized nanoparticle. As you can see, the toxo-loaded calcium carbonate mineralized nanoparticle have excellent and anti-cancer ability. The toxo-free control did not show any noticeable inhibition of tumor growth, and free dox suppressed at the tumor weight by about 6%, and the toxo-loaded mineral nanoparticle decreased the tumor weight by 5% compared to the cell line treated mice. Next research is about the photodynamic therapy systems. Using mineralized formulation, we successfully developed the photosensitizer loaded bubble generating mineralized formulation for ultrasound imaging and photodynamic therapy. So we also developed the nano container for the controlled topical delivery of antibiotics and treatment of bacterial biofilm. So by using this system, we also confirm the effect of doxycycline mineralized nanocontainer on biofilm formation and and on ultrastructure of bacteria. And you can see the figure, the nanocontainer show the most inhibition effect of biofilm formation and ultrastructure of bacteria. So, and next research is about the ROS reactive oxygen stress level controlled formulation study. As you can see, this picture, the level of ROS in the cancer is higher than the normal cell. It is the specific microenvironment point of cancer, and we can control the ROS amplify formulation to induce the apoptosis of cancer cell without the damage of normal cells. By developing of the ROS level controlled formulation, we can confirm the enhanced cancer therapy. So let's move into the next topic about the potential of metal carbonate mineral for cancer diagnosis. We can easily find many naturally mineralized crystalline structure: calcium carbonate having 3 crystalline polymorphs, lobosite from manganese carbonate, siderite from the ferrous carbonate are good examples. And we can classify additional carbonate biominerals. One of the bases of biomimetic mineralization, we have developed the various nano-sized mineralized nanoparticles and suggest their application as a contrast agent for MR-US multimodal imaging. As you know, the multimodal imaging is recognized as a tool for accurate tumor imaging by providing complementary and collective imaging information. There are many trials for nano-contrast agents for various multimodal imaging, such as MRCT and MRN PET and MRN ultrasound. Especially, we have focused on MR and US multimodal imaging. US MR is a complementary combination by combining advantages of each imaging modality. MR is characterized by high spatial resolution, whereas ultrasound is real-time imaging and has advantages of easy access for patients. So this system is mangan carbonate mineralized nanoparticle. Mangan carbonate is known to have the similar aqueous solubilization behavior of calcium carbonate minerals. At the natural pH, it retained its stable crystalline structure, but the low, slightly acidic pH, it can be ionized by generating T1, mangan ion, and carbonate gas molecules. As in the case of gadolinium mangan nanoparticle at the tumoral pH, mangan carbonate nanoparticle may simultaneously switch on of the MR and ultrasound signals. After reaching the extracellular matrix of the tumor tissues, they may display the US MR multimodal imaging functions by generating carbon dioxide microbubble and mangan ions. So this is the on and off system. So electron microscopy images show the morphology of mangan carbonate and the X-ray differential patterns and the spacing of the lactic verify the formation of rhodosite crystalline nanoparticle. So elemental mapping also show the elements which belong to the mangan carbonate mineralized nanoparticles. Now after making the multimodal contrast imaging agent, we can also confirm the T1 MR imaging and the ultrasound imaging also. As you can see, it showed the pH dependent release pattern, and as you exceed pH, the contrast enhancement is very higher than the normal pH, the image. So, by using this result, we are now working at the animal study. So at the last topic, I will introduce ongoing research at our lab. Recently, we are developing the cannabinoid delivery systems. As you know, the cannabinoid, one of the cannabidiol is one of the cannabinoid from the marijuana plant. It's also used for anti-tumor treatment. The mechanism is apoptosis of inducing the ER stress. After formulation of cannabidiol, I showed a very long sustained release pattern about 2 weeks. And we also confirmed the enhanced ER stress by using the Western blot. So the next ongoing research is about the mRNA vaccine delivery system. Now we are developing the formulation characterization. We are expecting this system show the good potential for mRNA delivery and vaccination. So I will summarize my talk today. I described the potential of mineralized nanoformulation. By this formulation technology, we can expand these systems to high-performance theranostic delivery systems. We have many templates and many ion species for mineralization and can induce the many therapeutic and diagnostic species into the mineralized formulation. If we have many ideas regarding how we combine these elements, we may generate the many high-performance systems for biomedical application. So we have been doing this work in collaborate with Professor Seong Jung at Gyeonggi University and Sang-Chun Lee at Gyeonggi University, and Ik-Chang Kwon at KIST and Kwang-Myung Kim at Ewha Women University. Especially, I want to special thanks to my members. So now my research project is recently the 4 category. The one is cannabis delivery system, and second is exosome delivery system using the microbiome, and also mRNA using the mRNA or siRNA. To develop the gene delivery system. So at the last, also I am working in the cancer immunotherapy. Yeah. Thank you very much. Thank you for your attention.